All right, let's get right into it. So back in early 2026, three different scientific discoveries pop up that, on their own were pretty fascinating. But what if they weren't separate at all? What if they were actually all just pieces of one enormous, planet-sized puzzle? Well, in this explainer, we're going to put those pieces together, and what they reveal is a kind of hidden engine running in our atmosphere. So you're probably wondering, what are these puzzle pieces? Well, we're talking about fungi that literally stole a gene from bacteria, trees that mysteriously start glowing during thunderstorms, and a newly discovered global network of microbes that are sharing information. I know, it sounds like something straight out of science fiction, but this is real cutting-edge science. And the connection between them? Oh, it's even more mind-bending than the discoveries themselves. Okay, so let's lay out the puzzle. We've got three discoveries that, on the surface, seem completely impossible and totally unrelated. First up, we've got a team led by Eufemio. They found that this common soil fungus, Morteria lacea, creates these incredible ice-nucleating proteins, or INPs for short. But here's the wild part. They didn't come up with this trick themselves. It turns out they snatched the gene from bacteria a long, long time ago. And they didn't just copy it, they actually upgraded it. They made the protein water-soluble and way more stable, which is a huge deal, because the original bacterial version had to be stuck to a cell membrane. That little detail is absolutely crucial. Next, we have McFarland and his team. They caught something happening for the first time ever out in the wild. It's called corona discharge, and it was happening on tree canopies. Basically, during a thunderstorm, the very tips of the leaves literally start to glow with these little bursts of UV light. You can think of it like a tiny natural neon sign that's being powered by the storm's own electric field. Just a totally bizarre and beautiful thing that nobody had ever documented in nature before. And a final piece of our puzzle comes from Schiller and Kim. They analyzed, get this, over 85,000 samples, and they discovered this small group of generalist microbes that act like little global couriers. They travel between totally different habitats. We're talking from soil all the way up to the clouds, and they're sharing genes the whole way. They're basically running a planet-wide biological internet, connecting ecosystems that we thought were totally isolated. So, just to recap, we've got superpowered fungal proteins, glowing trees, and the microbial internet. How on earth could these things possibly be connected? It seems impossible, right? But the source material we're breaking down today proposes this single incredibly elegant idea that ties it all together. And that missing piece, the concept that makes it all click into place, is a new framework called the needle-eye principle. So what exactly is a needle-eye? Well, imagine trying to squeeze something through a tiny, tiny opening. Whatever comes out the other side is changed, right? But it's still made of the original stuff. That's the idea. It's a bottleneck, a gate that acts as a transformer. It's a really simple concept, but trust me, it is the key that unlocks this entire mystery. So for something to be a needle-eye gate, it has to meet three specific conditions. First, there has to be a constrained or tight passage. Second, a transformation has to occur. The output can't be exactly the same as the input. And third, you have to be able to trace a clear connection between what went in and what came out. Keep those three rules in your back pocket as we go through this. All right, now that we have the needle-eye principle, we can finally start to assemble the puzzle. We're about to walk through a self-reinforcing 6-stage planetary loop that connects everything we've just talked about. This is the grand reveal. Here's the basic roadmap. It all kicks off with a storm, which then triggers the fungi, which then helps create ice in the clouds, which leads to rain, which then feeds the fungi, which then releases spores to start the whole thing all over again. It's a perfect circle. So let's break down each stage. Stage 1, it all starts with a thunderstorm. The storm's massive electric field is our input, and the tree canopy, that's our first needle-eye gate. The leaves and the branches constrain all that energy, transforming it into these focused little bursts of UV light happening right on the surface where life is. A huge diffuse energy field becomes tiny, concentrated hotspots. Now the rain from that storm soaks into the soil, and that's the trigger for our Morteria lacea fungi. And here's our second gate. Inside the fungus, that bacterial gene they stole is the input, and it's transformed into that super-powered water-soluble ice-making protein. The fungus itself is the biological needle-eye. And this stage, this is where you really see the power of a needle-eye. That one single protein molecule, now floating around in the atmosphere, bumps into a super-cooled water droplet. It acts as the perfect template. The perfect gate, transforming millions of chaotic liquid water molecules into a perfectly ordered ice crystal. A tiny, tiny input creates this massive structured output. It's incredible. So those ice crystals get bigger and heavier, and eventually they fall as rain or snow. In this stage, the entire cloud system is the gate. You have all this dispersed, invisible water vapor that gets transformed into concentrated, life-giving rain, which falls right back down onto the forest floor, watering the very same fungi that started this whole process. See? The loop is starting to feed itself. And this is where that third discovery clicks right into place. The now damp soil becomes this bustling hub of activity. Fungi and those generalist microbes start interacting, swapping genetic information. The soil habitat itself is a needle-eye gate, selecting which genes are actually useful and get passed around. It's like a tiny, natural genetic engineering lab. And finally, the fungi release their spores, coated with those amazing ice-nucleating proteins, back up into the air. Now the atmosphere becomes the final gate. It acts like a filter, selecting for particles that are just the right size, that are stable, and that are really good at making ice, which are precisely the traits that this fungal protein has evolved. The loop is now closed, all ready for the next storm to come along. Okay, so this whole loop is an amazing story about how nature works. But it also begs the question, can we learn from this? Can we actually apply this needle-eye principle ourselves? What happens when we try to engineer it in a lab? Well, it turns out we can. Scientists have developed this engineered system they're calling a pollen microbioreactor, or PMB, and it basically mimics these natural gates. Just like a fungal cell transforms a gene, these little bioreactors use tiny capsules to transform microbial metabolism. It's the exact same shared principle. You use a confined space, a needle eye, to control and transform whatever comes out. It proves we can learn from and engineer this fundamental idea. So, why does all of this matter? What's the big takeaway here? Well, the implications of seeing the world through this needle-eye lens are actually pretty profound. For one, this isn't just a cool story. The theory makes very clear, testable predictions. For example, scientists can now literally go out after a storm and check the air for these specific fungal proteins. They can build setups in the lab to see if corona fields actually make fungi release more of them. This is a real, falsifiable scientific hypothesis, and that's what makes it so exciting. And this all leads to the core takeaway, the big idea that reframes our whole perspective. Fungi aren't just passively sitting around in the soil, waiting for rain. It seems they might actually be active players manipulating the atmosphere to create the very conditions they need to thrive. They're part of a massive information system that connects life and weather on a planetary scale. And that leaves us with one last really provocative question. We tend to think of weather as this purely physical, non-living process. But what if this loop is real? What if, to some degree, the rain itself is a biological product, something that's shaped and encouraged by a vast, interconnected, living network? It's a thought that completely changes how you see the sky above you.